All right, let's dive right in and talk about one of the most amazing structures in all of biology, the plasma membrane. Honestly, it's the unsung hero of the cell. It's this incredible gatekeeper that defines the very edge of life itself, deciding what gets in and what has to stay out. So let's get a closer look.
I mean, really think about that for a second. When you get down to the microscopic level, what is the one thing that separates all the complex, amazing machinery of a living cell from, well, everything else? Yep, you guessed it, it's the plasma membrane. But here's the thing, calling it just a wall or a bag is a huge oversimplification. This barrier is anything but passive. It's active, it's intelligent, and it is incredibly dynamic. A much better way to think about it is like a super sophisticated, high-tech security system.
I mean, this membrane is a serious multitasker. It's got so many jobs to do. Of course, it separates the inside from the outside, but it also grabs signals from things like hormones, it carefully controls what comes in and out, and it even helps the cell move around and recognize its neighbors, which is crucial for building tissues. It's pretty amazing. And when you actually look at it under an electron microscope, wow, it's just this delicate, two-layered line. That's it. That's all that stands between the cell and the entire universe. To put that into perspective, a single sheet of paper is about 100,000 nanometers thick. This barrier is more than 10,000 times thinner than that, and yet it does all that incredible work. It's just mind-boggling. So how on earth do you build a barrier that's so versatile and so ridiculously thin? Well, it all starts with one very special, very clever little molecule. Let's take a look at how this whole foundation gets put together. And that leads us to this really fascinating chemical puzzle. We all know the old saying, right? Oil and water don't mix. And the environment both inside and outside of a cell is, mostly water. So how is it possible for a membrane, which is basically an oily, fatty structure, to even exist in the first place? The secret is this absolutely brilliant chemical trick. The molecules that build the membrane aren't just oily, they're what we call amphiphilic. It's kind of like they have a split personality. One part of the molecule absolutely loves water, and the other part is terrified of it and will do anything to get away. And here is the star of the show, the phospholipid. You can see that split personality perfectly. You've got this round head, which is hydrophilic. It loves hanging out with water. And then you have these two long tails, which are made of fatty acids. They're hydrophobic. They hate water. This super simple design, well, it's the secret to everything. Okay, but the membrane isn't just made of these phospholipids. They're the foundation, for sure. They create this kind of oily sea, but there are a bunch of other essential players embedded within that sea, and all together they create what scientists call a molecular mosaic. If you look at what the membrane's made of by weight, it's really interesting. Lipids, our phospholipids, make up about half of it. But look at that, proteins are almost just as big a component. And these proteins are the real workhorses. They're the channels, the pumps, the receptors that are doing all the communicating and transporting. And those little carbohydrate bits, think of them like ID tags on the cell's surface, helping it talk to the world. Now, let's zoom back in on those fatty acid tails for a second, because there's a really important detail here. Some of them are perfectly straight. We call those saturated. But others have a little bend or a kink in them. Those are called unsaturated. Now, this might seem like a tiny, tiny detail, but trust me, it has massive consequences for the cell. You can see it so clearly here. The saturated one is just a straight line. But in the unsaturated one, that double bond physically forces a rigid kink into the chain. Just imagine trying to stack a neat pile of straight logs versus trying to stack a pile of crooked branches. The crooked branches are never going to pack together as tightly, right? They'll always leave more space. Just keep that in mind. And there's one other key player we have to mention, cholesterol. This guy wedges itself right in between the phospholipids. Its job is to be a fluidity buffer. When it gets cold, it keeps the tails from packing too tightly and freezing solid, and when it gets hot, it keeps them from getting too loose and falling apart. It keeps everything just right. So let's put it all together. You've got this sea of phospholipids, and floating around in it are these big protein icebergs with little cholesterol molecules tucked in between them. Then on the outside surface, you have these carbohydrate chains sticking out like antennas. This whole beautiful, complex picture is what we call the fluid mosaic model, an idea that totally changed how we understood cells back in the 70s. Okay, so we've talked about the mosaic part, all the different pieces fitting together. But now for the part that I think is just going to blow your mind, the fluid part. This entire structure is not solid, it's not static,
It is constantly moving, shifting, and rearranging itself.
The individual phospholipid molecules are constantly just scooting past each other, drifting sideways, kind of like people mingling in a really crowded room. They do this all the time, and they do it fast. But what they almost never ever do is flip-flop over to the other side of the membrane. Why? Well, that water-loving head would have to travel through the water-hating core, and it just does not want to do that.
And that's why this quote just nails it. It really isn't a wall at all. It actually has the consistency of something like olive oil or salad dressing. And those big proteins, they're not locked in place. They're just like little boats floating around in this two-dimensional oily ocean. And this is where those little kinks in the fatty acid tails make a huge comeback. Remember how they keep the phospholipids from packing together too tightly? Well, that's what makes the membrane fluid. And this fluidity isn't just a cool feature, it is absolutely essential for life. It allows proteins to move around and bump into each other, which is critical for sending signals, and it lets the entire cell change its shape, move, and even divide in two. So the crucial point, the big takeaway from all this, is that when you picture a cell, please don't picture a simple static bubble. Picture this instead, a constantly shifting, incredibly complex, intelligent sea of lipids and proteins, actively sensing its world, controlling everything that goes in and out, and defining that sacred little space that we call life. It's just such an elegant structure when you think about it. You have these very simple rules of chemistry, water-loving heads, water-fearing tails, and from that, you get this stunningly complex biological machine. It really makes you wonder, doesn't it? What other amazing marvels of life are built on foundations that are just as simple but just as profound?